The next topic in chapter 14 is the regulation of blood volume. By weight, the human body is about 60% water. 2/3 of this water is within cells, so it's within the intracellular fluid compartment. The other third is outside of cells, so it's in the extracellular fluid compartment, or ECF. And then if we look at the ECF, within that, about 20% is blood plasma, and then the other 80% is interstitial fluid. Most of the time, the amount of water in the various compartments is relatively stable. So this condition of relative stability is called dynamic steady state. And the reason you have this dynamic steady state is you have a number of homeostatic mechanisms, which we'll talk about, that help to maintain the composition of the extracellular and intercellular fluid compartments stable. So in this diagram in the middle, we can see that you have an extracellular environment. And so there's essentially flow of water between, and solutes and things like that, between the extracellular environment and the extracellular fluid, or ECF, and vice versa. And then from the extracellular fluid, there's movement of water and solutes between ECF and the ICF, or intracellular fluid. And we'll talk about that in more detail in a second, using the example of drinking a glass of water. So you can see this picture, this woman's drinking a glass of water. So this is part of her water intake. So she also, of course, also has water in the food that she eats. And recall that you also make water by aerobic metabolism. Recall that oxygen is the final electron acceptor in the electron transport chain. And together with that electron comes hydrogen. And so you have the formation of H2O, or water, that way. That's a relatively minor component, but it still is part of the water that you can count as intake. And then in terms of water output, you have water, of course, in urine. So we'll talk a lot about the formation of urine. So humans have the ability to make very concentrated urine, which allows us to live in desert environments. We can also make quite dilute urine, and so that helps in the case we say to drink too much water. We can excrete the excess water. Now, we constantly lose water as water vapor in our lungs. So within our lungs, we have a core temperature of 37 degrees centigrade, and at that temperature, we have the airspace within the lungs is fully saturated with water, meaning it has 100% relative humidity. So as you breathe out, then there's quite a bit of water vapor in that. There's also water vapor, there's also water within feces, and of course we lose water as well as salts with sweat. Let's follow the water that this woman was drinking and see how it ends up in the body. So we'll start with water intake. So she's drinking water. So we have water entering in via the small intestine. It's going to enter into interstitial fluid. And from there, it'll enter into capillaries that are associated with the small intestine. So this will actually increase the blood volume. It'll be subtle, but every time you drink, there's a small increase in blood volume. That doesn't last very long because, of course, the water gets distributed quite quickly. So the blood from in the capillaries will then move to smaller into venules and veins and so forth, and enter into the general circulation. And from there, it'll move into the interstitial fluid. So water moves quite readily. Remember that most capillaries are a little bit leaky, particularly for small solutes in water. So the water will enter into the interstitial fluid, and the water will actually move by osmosis then into cells. So the cell volume will actually increase. So this, again, is a subtle effect, but if you drink a glass of water, you will, in fact, slightly increase your blood volume, and then a short while after that, increase the volume of your cells. Now, of course, there's also loss of water. So in the kidney, we have filtration. And in the filtrate, you have water and solutes, and that filtrate eventually becomes urine. Now, we do a lot of reabsorption of that filtrate. So we bring salts and water back in, and we'll talk about that in a second. But there is water loss, of course, in urine. As I mentioned earlier, you have water vapor loss across lungs, and of course you have water loss via sweat, and of course it's not shown here. Oh, it is shown here. Feces, you have some water loss there. As I mentioned earlier, the distribution of water between the various fluid compartments of the body, for example, the intracellular fluid, interstitial fluid, and blood plasma, is in a state of dynamic steady state. This means that overall, normally when you're healthy and so forth, the values don't change very much, but it's dynamic. It can change, and it is changing a little bit over time. So one of the first things we need to understand is what happens at the capillaries. So this is referred to as the tissue capillary fluid exchange. And so if we look at a capillary, we'll have an arteriolar end, which is the end that's closer to the heart, where the left ventricle and the aorta and so forth. So blood is moving in this direction. And then you have a venular end, which is the end that in which blood will go into the venules and then into the veins and end up in the right atrium. So blood is flowing in one direction through the capillary. As this occurs, there is loss of fluid via a process called fluid filtration across the walls of the capillaries. Recall that most of the capillaries in your body do have some small clefts between the cells. Even in capillaries that have tight junctions, often there are small places, areas where there are in fact little pores or clefts between the endothelial cells. And these clefts will allow for the movement of water as well as solutes, such as sodium, chloride, potassium, magnesium, calcium. Also, it'll allow for movement of some organic molecules such as glucose and so forth. So there's a continual movement of water and these smaller solutes through these small gaps in the capillary walls. Note that proteins and cells do not move. So red blood cells are confined within the capillary, and proteins, which are relatively large and also negatively charged, are prevented from passing through these pores. Now, they technically could get through the pores. They're small enough they could get through the pores, but there's another layer called the basement membrane, and that prevents the movement of proteins through these leaky capillaries. So the fluid that leaves these capillaries is called the filtrate. So here we can see this fluid leaving. So this fluid, once it's in the interstitial space, is called the filtrate. And you can also refer to it becomes the interstitial fluid. So we'll also refer to that as the interstitial fluid. And what's being shown here is that the movement of this fluid is driven by blood pressure, also called hydrostatic pressure. And what you note with these arrows is that the arrows get smaller and smaller as you move from the arteriolar end to the venular end. And the reason for that is that the blood pressure or hydrostatic pressure decreases as you move through the capillary. And there's two reasons for this. One is it's simply the fact that the capillary has resistance to flow. And so as the fluid moves through a capillary, there's friction between the moving fluid and the walls of the capillary that generates heat. And so there's loss of this pressure, which can be thought of as a potential energy. It'll be turned into heat. But then the other thing is you can see in this picture is there's fluid leaving. And so this means the volume of the fluid is going down within the capillary because there's less and less fluid. And that also affects the pressure significantly. So that's another reason. As the fluid leaves, it actually changes the pressure within the capillaries. The next thing we're going to do is calculate the hydrostatic pressure difference between the fluid within the capillary and the fluid outside of the capillary. So the hydrostatic pressure in the capillary is P sub C. So it stands for pressure within the capillary, and that's 37 millimeters of mercury. The hydrostatic pressure of the interstitial fluid, or P sub I, is 1 millimeter of mercury. Note that it's very low in the interstitial fluid, and the reason for that is that you're basically normally you don't allow for fluid to build up in the interstitial space. You have fluid there, but there's the lymphatic system that helps to drain this excess fluid out. Now, there are occasionally situations where this may increase. There's disease states and so forth where it might increase a bit. But we can then calculate the net filtration pressure, simply the difference between these two. So PC P sub C minus P sub I. So we have a net filtration pressure of 36 millimeters at the arteriolar end. Okay, now let's look at the other end, at the venular end. We have, at this point, a hydrostatic pressure in the capillary, or P sub C, of 17. So now this dropped pretty significantly from 37. And the hydrostatic pressure of the interstitial fluid is constant, so it's about 1 millimeter of mercury. So then our difference is that the net filtration pressure is now 16 millimeters at the venule end, millimeters of mercury. Recall that within blood you have, in addition to red blood cells and white blood cells, you also have solutes, but also proteins. And as I mentioned earlier, within the filtrate, you have water and solutes, such as sodium chloride, potassium, and glucose, and things like that. But there's a real absence of protein. And so there are some proteins in the interstitial fluid, but the amount of protein is much less. And the reason for that is because proteins are simply don't pass through the capillary into the interstitial fluid. And so if you look at the concentrations of solutes like sodium and chloride and so forth, they're very similar between the blood plasma and the interstitial fluid, but the protein concentration is much, much lower in the interstitial fluid. And that's illustrated in this picture here. You can see there are proteins which are represented as these yellow circles. And it's also pointing out here on this diagram that some of these proteins are albumin. So the majority of these proteins are what's called serum albumin. That's the most abundant protein in human blood plasma. Now there are other proteins too. So imagine there's a number of these proteins here. Well, what effect will that have on the osmotic movement of water? Well, remember, water moves down its concentration gradient. So these proteins take up space. And so this means that water will move from an area of high water concentration, which is in the filtrate or the interstitial fluid, back into the capillaries. And that's being shown here, these 3 arrows. So we would expect fluid filtration in one direction because of the hydrostatic pressure difference, and then we have fluid flow in the other direction back into the capillaries due to this osmotic movement of water. And so this again, this protein concentration of the interstitial fluid is lower than the protein concentration of blood plasma. The colloid osmotic pressure, or pi, is the osmotic pressure due to proteins. This is also called oncotic pressure. So we have this sort of overall osmotic pressure of a fluid. But if we can separate out and just look at the component of that osmotic pressure that's due to the presence of proteins, we can do that, and then that's called the osmotic pressure or oncotic pressure. And it turns out that this colloid osmotic pressure, or pi, is higher within the capillaries than within the interstitial fluid. So pi sub p is greater than pi sub I. And we can look at that difference. So typically that difference is going to be about 25 millimeters of mercury. And so this pressure difference drives the osmotic movement of water into the capillaries. The combination of hydrostatic pressure and oncotic pressure predicts the movement of fluid across the capillary membrane. All this was originally determined by Starling. And so these forces, when you combine them all together, are referred to as the Starling forces. So fluid movement is proportional to fluid out, forces that push fluid out, and minus the forces that push fluid in. So let's look at these. So what forces would lead to the movement of fluid out? Well, the hydrostatic pressure within the capillary would tend to move fluid out, and the colloid osmotic pressure of the interstitial fluid would tend to move fluid out. Let's look at forces that move fluid in. The hydrostatic pressure of the interstitial fluid would tend to force fluid in, and the colloid osmotic pressure of the plasma, which is pi sub p, would tend to draw fluid into the capillary. So therefore, the overall net pressure, or P sub net, is equal to the difference between these, as you can see in this equation here. We're now in a good position to now calculate the net pressure at the arterial end of the capillary and at the venous end of the capillary. And so taking into account all the Starling forces that we just discussed. So at the arteriolar end of the capillary, P sub net is positive 11 millimeters of mercury. So this means that there's a pressure, net pressure, that would lead to the movement of fluid out of the capillary into the interstitial space. At the venous end, the net pressure is negative 9 millimeters of mercury. So this means there's an overall net pressure that pushes fluid from the interstitial space back into the capillary. So we can now introduce two more terms. This means that the arteriolar end, there's going to be net filtration due to this pressure difference, and so the fluid is moving out. At the venous end of the capillary, you have a net negative filtration pressure, and so we have net absorption, sometimes referred to as net reabsorption. But this means that fluid is now going to move from the interstitial space and back into the capillary. In this diagram, we can see that there's net filtration at the arteriolar end of capillaries and at net absorption at the venous end. Of course, this filtration and absorption is driven by the Starling forces, which we discussed earlier. The return of fluids on the venous end is not 100%. In other words, 10 to 15% remains in the interstitial spaces. And this fluid will ultimately return to the blood plasma through the lymphatic system, starting with lymphatic capillaries, going to lymphatic vessels, and then lymphatic ducts. So about 1 to 2 liters of interstitial fluid per day enters the lymphatic system. And so this is fluid that's essentially recycled. It comes back into the blood plasma each day. Changes in the Starling forces can lead to edema, a condition in which there's excessive accumulation of interstitial fluid. This may be the result of high arterial blood pressure. If you think about it, this will increase the capillary pressures, and this will lead to an increase in net filtration pressure, leading to an increased filtration. Another thing that could cause edema would be venous obstruction. This may happen, for example, in the superficial veins in the legs, and that's a condition called thrombophlebitis, in which inflammation starts, and then there's often blood clotting that goes with the inflammation, and this could lead to venous obstruction. So this essentially would lead to back pressure in a sense. The blood isn't flowing properly through the capillaries, and therefore there'll be an increased pressure again within the capillaries, and this would increase the net filtration pressure, leading to an increase in net filtration. Inflammation and allergic reactions may result in an increase in capillary permeability to proteins. Therefore, there will be leakage of plasma proteins into the interstitial space. This will increase the colloid osmotic pressure of the interstitial space, which is normally very low. Water will follow osmotically, and therefore there will be increased net filtration of fluid out of the capillaries. And this leads, of course, to the increased swelling and so forth around an area where there's inflammation or an area where there's been an allergic reaction. There are a number of other conditions that can lead to edema. One is myxedema. This is another term for severely advanced hypothyroidism. In this condition, there's an increase in production of mucin, which is a glycoprotein, which is a constituent of mucus. This protein is produced in the extracellular matrix, and it leads to an increase in the colloid osmotic pressure of the interstitial space. So as this increases in the interstitial space, this leads to less reabsorption of water into the capillaries, and therefore that causes edema. Another possibility is a decreased plasma protein concentration. So most of the plasma proteins are created by the liver. So in situations of liver disease, there may be a decrease in the plasma protein concentration. So this reduces the colloid osmotic pressure within the capillaries, and this leads to less reabsorption of water across the capillary walls. In addition to liver disease, kidney disease may also lead to a change in plasma protein concentration. Normally, filtration in the kidney works such that blood cells as well as proteins are left within the blood plasma and are not filtered. In other words, they don't end up in the filtrate within the kidney tubules. But in certain disease states, these proteins are actually able to enter into the filtrate. And so that would lead to a decrease in plasma protein concentration, and that would have lead to a decrease in colloid osmotic pressure of the fluid within the capillaries, and this would lead to less reabsorption, and therefore there would be edema. Finally, there can also be obstruction of lymphatic drainage. So when this happens, there'll be a buildup of the interstitial fluid because it's not able to enter the lymphatic capillaries and become lymph. And so this ends up leading to edema. There's two different types of common types of obstruction. One has to do with the parasitic nematode worm. These will infiltrate the lymphatic system and they'll end up blocking the lymphatic system and therefore drainage. And second type that is also fairly common is after surgery. So for example, for cancer surgery, there may be removal of lymph nodes, and this may cause some scarring and things like that, or inoperable ducts and so forth. And therefore, this can also lead to obstruction of the lymphatic drainage, and this can also lead to edema. Filariasis is a tropical disease in which blood-sucking insects such as mosquitoes spread a parasitic nematode worm. And if this condition is allowed to go on far enough, it becomes elephantiasis, as you can see here, in which you have extreme swelling. In elephantiasis, the species of worms takes up residence in the lymphatic system, and the larvae block the lymphatic drainage. This disease is found in about 72 tropical countries, where over 1 billion people live, and these people are threatened by infection. There is, luckily, effective drug therapy against this parasite. This table summarizes the various causes of edema. You may have seen pictures of starving children that have very swollen abdomens, and you may have wondered why they had such swollen abdomens. This is a sign of protein malnutrition. We've learned that a decrease in plasma protein concentration leads to less osmotic movement of water from tissue in the interstitial fluid back into capillaries. This then leads to edema. So it turns out in certain countries where they have a poor quality diet with not enough protein, this can then lead to very low protein levels within the blood and then edema. So the swollen abdomen is in fact a symptom of this very poor diet, which with very little protein. And it actually occurs typically in a child when the second child is born and is now being breastfed. So in other words, when a child is born, it then receives breast milk, and that breast milk is high in protein and very good, and there's good growth and so forth. As soon as the second child comes along, then the mother starts essentially weans the first child, and the second child now receives the milk with the high protein and so forth. And so you start to see then these symptoms in the first child. We're next going to look at the regulation of blood volume. It turns out that blood volume is extremely important because it determines blood pressure. So if there's more fluid within the vascular system, then that fluid then can push on the walls and increase the blood pressure. And recall that mean arterial pressure is extremely important. In other words, it's a regulated variable. Our body pays a lot of attention to it, and it has homeostatic mechanisms that work via negative feedback loops to conserve or keep the mean arterial pressure within a very narrow range. So tied in with maintaining mean arterial pressure is regulating blood volume. So your body will also have a lot of mechanisms that help us regulate blood volume. One of the main organs that's involved in blood volume regulation is the kidneys. The formation of urine begins with with filtration of fluid through capillaries, and the kidney is called glomeruli. So, for example, here you see blood in a capillary within the kidney. It goes into this knot of small blood capillaries called the glomerulus, and then the blood exits. Now, due to a lot of the Starling forces we just talked about, there is the generation of filtrate. The filtrate ends up in the lumen of a structure called the Bowman's capsule. From there, it goes into the renal tubule. And so that filtrate will contain water, and it will contain solutes, but it won't contain blood cells, and it won't contain very much protein. In fact, very little protein. So normally in a typical person, you have about 180 liters of filtrate produced per day in your kidneys. But in a typical person, you only generate about 1.5 liters of urine. So this means that most of the fluid is in fact reabsorbed back into the blood, and that's shown here during step 2 here, reabsorption. You have the reabsorption of water, and you also have the reabsorption of solutes, such as sodium and chloride. The amount of fluid reabsorbed is controlled by several hormones and the sympathetic nervous system, and this all occurs in response to the body's needs. Let's first look at the role of the sympathetic nervous system on blood volume regulation. Imagine there's a lot of venous return to the heart. This will increase the blood volume in the atria. There are mechanosensitive receptors in the atria that respond to that stretch by becoming more active, and there are action potentials that are sent via the nervous system to the brainstem. This leads to an increase in sympathetic stimulation of the heart, which results in an increase in heart rate and heart contractility. And the reason for all this is we don't want blood to pool in the heart, and so by having increased heart rate and contractility, this tends to keep the blood moving along nicely. Now, the second thing that happens with the activation of these atrial stretch receptors is there's actually going to be a decrease in sympathetic stimulation to the kidneys. So normally what sympathetic stimulation does for many of the blood vessels in our body is it leads to a sort of tonic vasoconstriction. So in other words, tonic means it's an ongoing vasoconstriction. So if there's decreased sympathetic stimulation of the kidneys, this means the arterioles within the kidney will dilate. Arterioles are, remember, the small blood vessels that are encircled by smooth muscle right before a capillary bed. This will lead to increased blood flow into the capillary beds, the glomeruli within the kidney, and this will lead to increased filtration and therefore increased urine production. And this then causes a decrease in blood volume, and that, of course, opposes this increased venous return and increased blood volume that the heart was detecting via these mechanosensitive receptors, stretch receptors within the atria. One of the major hormones that regulates blood volume is antidiuretic hormone, or ADH. It's also called vasopressin, and sometimes it's also referred to as arginine vasopressin because that's the form that mammals have. ADH is produced by the hypothalamus and released from the posterior pituitary when osmoreceptors, which are within the hypothalamus, detect increased blood plasma osmolality. So why would blood plasma osmolality increase? It could be due to excessive salt intake or dehydration. So one of the effects of this activation of these osmoreceptors in the hypothalamus will be increased thirst. So a person would drink, and of course that would help to dilute the blood and decrease the osmolality of the blood. The other thing that could happen is you could try and retain water within your body, and that's the role of ADH. So ADH is, as you can see in this diagram, it's produced by these neurosecretory neurons within the hypothalamus. It's transported down the axons. It's then released at axon terminals into capillaries within the posterior pituitary. It then joins the general circulation. Within the kidney, there are receptors for antidiuretic hormone. And what will happen is that when these receptors are activated, is you'll have an increased expression of aquaporin channels in the plasma membranes of certain cells that make up the walls of the collecting ducts within the kidneys. And this will then lead to more water reabsorption through these aquaporin channels. So the water will exit from the filtrate, which was within the lumen of this tubule, and end up in the interstitial space. From there, it can enter into capillaries within the kidneys and enter back into the general circulation of blood. So to recap, ADH will stimulate water reabsorption from the filtrate. That helps with increasing blood volume. Of course, thirst, which is also stimulated by these osmoreceptors in the hypothalamus, also helps with increasing blood volume. So those two things together are often sufficient to bring a person back from a state of dehydration. So as I just explained, an increase in water intake and decreased urine formation. In other words, you're making much more concentrated urine when there's ADH available. This will increase blood volume. So this will end up making the blood more dilute, and then at that point, ADH secretion will decrease. And so this is all a negative feedback loop in which ADH secretion is controlled in a way that helps to maintain blood osmolality. Now, it turns out your body also pays attention to pressure. There are stretch receptors in the left atrium, in the carotid sinus, and in the aortic arch. And these actually will inhibit ADH release or secretion. And that makes sense because remember, ADH is released typically in situations where the blood has become more concentrated. There's an increase in osmolality. In a situation in which, and often together with that, there's a decrease in blood volume. In a situation, in the opposite situation, when a person is well hydrated, there'll be an increase in blood volume. And so that will increase the stretch of these stretch receptors. And therefore, basically the body's telling the system that we don't need as much ADH to be secreted because the blood volume is fine. Stretch receptors in the atria also stimulate the release of atrial natriuretic peptide, which increases excretion of salt, sodium chloride, and water from the kidneys to reduce blood volume. So this is essentially an antagonistic hormone to antidiuretic hormone. So antidiuretic hormone release leads to the reabsorption of water and therefore an increase in blood volume. Atrial natriuretic peptide does the opposite. It is stimulated when there's a high venous return and therefore a stretch of these atrial receptors, and it leads to the excretion of salt. Now, together with the excretion of the salt, the sodium and chloride, there'll be osmotic movement of water across the kidney tubules such that there'll be more dilute. Well, the water will move together with the salt by osmosis, and therefore the kidneys will produce more volume of urine. And then this will, of course, decrease blood volume. And a decrease in blood volume, remember, is tied into pressure. So there'll be a decrease in the mean arterial pressure. So atrial natriuretic peptide will decrease blood pressure. As I mentioned earlier, the control of blood volume by ADH is an example of a negative feedback loop. And we can look at that in more detail here in this figure. So we start with an initial stimulus. We have a guy in the desert. He forgot to bring water. It's very hot. And so he's dehydrated. And so he'll have a decrease in blood volume. Perhaps he's tried to eat some salt. I don't know. There's some salt ingestion going on also. And either of these factors or both together would then lead to an increase in blood osmolality. So his blood is essentially getting more concentrated. Okay, so now this will then stimulate osmoreceptors, which are neurons within the hypothalamus. They will then activate thirst. So if he's able to drink, he'll drink. This will, of course, increase the blood volume and decrease the blood osmolality. At the same time, within the hypothalamus, you have neurons that are neurosecretory. They'll be stimulated, and from axon terminals within the posterior pituitary, they'll release antidiuretic hormone into the general circulation of the person. This will then activate receptors in the kidney, and it'll lead to water retention. So the person will generate more concentrated urine and retain the water in the blood plasma. So this again also helps with increasing blood volume and decreasing blood osmolality. Recall that atrial natriuretic peptide, or ANP, causes the excretion of sodium and chloride in the kidney. So water osmotically follows the sodium and chloride. This means that ANP also causes an increase in urine volume, and this increase in urine volume causes a decrease in blood volume. Therefore, ANP decreases blood pressure. Now, a hormone that does the opposite, has the opposite effect, is aldosterone. This is a steroid hormone secreted by the adrenal cortex, which you can see here. The cortex is the outer layer of the adrenal gland. The adrenal glands sit on top of the kidneys. This hormone stimulates the reabsorption of sodium and chloride in the kidney. So sodium and chloride end up in the filtrate together with other solutes such as glucose. Sodium and chloride are reabsorbed. As they're reabsorbed, water follows osmotically. In other words, the filtrate becomes more dilute, and there's therefore a higher water concentration in the filtrate compared to the interstitial fluid, and water then moves down its concentration gradient. And so this is the osmotic movement of water from the lumen of the kidney tubule and into the interstitial fluid. And then from there, the sodium and chloride as well as the water end up in the capillaries, and then it joins the rest of the blood plasma eventually. So aldosterone essentially has the opposite effect as atrial natriuretic peptide. Aldosterone will increase blood volume, and as a consequence, it will increase blood pressure. So here's some more facts about aldosterone. The secretion of aldosterone is indirectly stimulated by salt deprivation and by low blood volume and by low blood pressure. It stimulates the reabsorption of salt and water in the kidneys. Aldosterone does not change blood osmolality, since the reabsorption of salt causes the passive reabsorption of water by osmosis. And aldosterone is regulated by the Renin-angiotensin-aldosterone system, or RAS. The renin-angiotensin-aldosterone system is stimulated by low blood pressure. The low blood pressure causes a decrease in blood flow to the kidneys, and there is something called the juxtaglomerular apparatus in the kidneys, which in response to this low blood flow will release or secrete an enzyme called renin. Now, renin is important because it converts the molecule angiotensinogen, which is a precursor to angiotensin 1, to angiotensin 1. Okay, so this is the ogen refers to this molecule being a precursor. It now becomes a hormone. Now, angiotensin 1 is then converted to a much more active form of the hormone called angiotensin 2 by an enzyme called ACE. ACE stands for angiotensin converting hormone. It's mainly located in the capillaries of the lungs. Angiotensin 2 has many effects that result in a rise in blood pressure. Angiotensin 2 causes vasoconstriction of small arteries and arterioles to increase the total peripheral resistance. It also stimulates thirst centers in the hypothalamus. That will, of course, increase blood volume. And thirdly, it stimulates the adrenal cortex to produce aldosterone. So recall that aldosterone will stimulate the kidney to reabsorb sodium and chloride. And with that reabsorption, you have the osmotic flow of water, and that will then also increase the blood plasma volume. So this pathway, as you can see, its purpose is to increase blood pressure. And therefore, it's not surprising that ACE inhibitors And angiotensin receptor blockers or antagonists of these angiotensin receptors can reduce blood pressure, and these are both used to treat hypertension. So aldosterone tends to lead to the reabsorption of sodium chloride, and you also have the osmotic flow of water back from the filtrate and back into the blood plasma, and this effect would then increase blood pressure. Now, the opposite effect we already established can be achieved through atrial natriuretic peptide, or ANP. This peptide is produced in the atria of the heart when stretch is detected from high volume of blood or by increased venous return. Atrial natriuretic peptide, or ANP, promotes salt and water excretion in the urine in response to this increased blood volume. In addition, the stretch receptors in the left atrium send sensory information to the hypothalamus, and they inhibit antidiuretic hormone secretion, or ADH secretion. And so this makes sense. The aldosterone increases blood pressure, ANP decreases blood pressure because it's stimulated by the stretch of the atria. I mean, its secretion is stimulated by the stretch of the atria. At the same time, antidiuretic hormone, when it's present, it tends to increase blood volume by increasing reabsorption of water. So the inhibition of ADH secretion should lead to less reabsorption of water within the kidney, and therefore more dilute urine when larger volumes of urine, and therefore a decrease in blood plasma volume. So in many ways, atrial natriuretic peptide acts as the antagonist, physiological antagonist of aldosterone, has all exactly the opposite effects. And in fact, there's another effect here, which is it shows here in this diagram that it also atrial natriuretic peptide seems to relax vascular smooth muscle in arterioles and venules, and that would also tend to decrease blood pressure. Although atrial natriuretic peptide, ANP, and aldosterone are functionally really antagonists, more so than antidiuretic hormone and ANP, antidiuretic hormone and ANP also have essentially opposite effects, although slightly different. Antidiuretic hormone, recall, doesn't have any effect on sodium or chloride excretion or reabsorption. It only affects water reabsorption. And yet it does act in many ways as a functional antagonist of ANP. And we'll see how this works in this diagram here. So we're going to take a case where we have high blood volume and or high venous return. And usually those go together, but you could have, for example, high venous return, for example, when you're exercising or something that without a particularly high blood volume. So when you're exercising, the skeletal muscle pump in the legs is working well, the respiratory pump is working well. So you may have high venous return without a particularly high blood volume. Often these two go together. These lead to an increase in ANP, atrial natriuretic peptide secretion, and a decrease in ADH, or antidiuretic hormone secretion. So let's look at this. And this is an example too. If you're in water, that tends to increase the venous return, even though you might not have a particularly high blood volume. The reason is normally if you're in an upright position, you'll have a lower venous return because the blood has to fight gravity to get back to the heart. Now, if you're in water, it's almost as if it's an anti-gravity sort of force. You're in the water, and it's much easier for blood to make it back to the heart, particularly if you're in a horizontal position, as demonstrated here by this child. And so, basically, if you're a child like this and you're in the water, this is going to increase urine production, as we'll see as we look at this. So we have water immersion, which would increase the venous return, or we could have an increased blood volume for whatever reason. Now, both of these things will lead to an increased venous return. This will lead to a stretch of the left atrium. Now, stretch of the left atrium will lead to an increase in the secretion of atrial natriuretic peptide, or ANP, and this leads to the stimulation of more excretion of sodium and chloride at the level of the kidney. And water follows the sodium and chloride osmotically, and so you'll also have more water excretion. So you'll have essentially more volume of urine produced. So this is shown here. There's an increase in urine volume. And if you have an increase in urine volume, then you'll have a decrease in blood volume. And so this forms a negative feedback loop. So as the blood volume goes down, then the venous return will go down, and there'll then be less stretch of the left atrium. So this is a very good example of a negative feedback loop. Now let's look at what happens on this other pathway. So the stretch of the left atrium will also, via the vagus nerves, send impulses to the brain. And the brain will send a signal to the posterior pituitary. So technically, when it says brain here, we're talking about the hypothalamus, and we have neurosecretory cells that produce antidiuretic hormone in the cell bodies. It's transported down their axons, and it's released at axon terminals in the posterior pituitary onto capillaries within the posterior pituitary. Now this ADH is now in the circulation, but the effect here is that with the stretch of the left atrium is that there'll be a decrease in antidiuretic hormone secretion. And this makes sense because normally antidiuretic hormone helps the kidney to reabsorb more water. So now there'll be a decrease in water reabsorption in the kidneys, meaning you'll make more dilute urine, and you'll make a larger volume of urine. So again, we have an increase in urine volume. This decreases blood volume, and then this leads to a decrease in venous return, and therefore less stretch of the atrium. So it's, again, that's also a negative feedback loop there. And so this also explains why maybe you don't want to swim in the shallow pool with all the children, because they're going to naturally be producing a lot of dilute urine due to what you can see here in this slide. As you exercise, you will lose water and electrolytes due to sweating. And this will lead to a lower blood volume, a lower cardiac output due to the lower venous return, and therefore the lower stroke volume. This can also have a negative effect on the ability of the body to dissipate heat. You need proper blood flow through the capillaries of the skin to dissipate heat properly. So you can alleviate some of these problems by drinking water. This can increase blood volume a bit, but one of the problems is if you just drink water without any electrolytes, and primarily we're talking about sodium, potassium chloride, which are lost in sweat, then this will change the osmolality of the blood. And this can be problematic. So sports drinks contain electrolytes, and they often also have sugars that can improve physical performance. As you're exercising, you use up the glucose in your blood, and you'll also end up breaking down glycogen in the liver, which will be released as glucose in the blood. So in order to replenish those glycogen sources within the liver, as well as actually glycogen within the muscle, it's good to drink something that, in addition to electrolytes, also has sugars. And here's an example of one. It's not the name is not all that enticing. It's a Japanese drink, sports drink called Pocari Sweat. But scientifically, it makes good sense, even though the name may be a bit of a marketing disaster if they tried to sell it in the United States. But essentially, it contains the sodium, potassium, and chloride in your sweat, as well as some sugar. And so it's quite a good sports drink. And actually, it's very popular in parts of Asia.